Good morning, everyone, and thank you again for being here with us. For me, it's a pleasure to introduce you, Professor Carlos Simón, my friend, my colleague, my mentor, and he's going to develop a little bit about the recurrent implantation failure of the endometrial factor. So, Carlos, please, whenever you want, the floor is yours.
Thank you, Nasser. Well, recurrent implantation failure exists, and you know, the human species is quite inefficient in terms of reproduction. We know that in a sunny day, implantation rate will go for 35%. We know that with euploid embryos, we can go to 60% implantation rate. But there are patients that they don't even achieve this bar, and they need more cycles to become pregnant. And this is what we call recurrent implantation failure, the failure of the process to go ahead. And then for that, clearly we have the main two factors. The embryo is always there, is the star, is where everybody is watching on. And of course, we need that the embryo will be euploid. This is quite obvious. I think nowadays, to transfer an embryo that is not already diagnosed as euploid, I mean, it will be a loss. But once the embryo is euploid and is being transferred, we know again that we can achieve this 60% pregnancy rate, but no more than that. And therefore, the endometrial factor exists, and exists even more in those patients that fail the endometrial factor, such as those of recurrent implantation of endometrial origin. So, in order to just turn our eyes to the mother and to see the endometrium itself, what we have learned in the last 10 years is that, first of all, there is an endometrial microbiome that matters, that a dysbiosis in the endometrial microbiome could affect implantation, and second, that there is a normal status, which is the initial addition part of the embryo, which is endometrial receptivity, that is also a personalized matter that altogether makes that personalization, which is always what we think on, may improve the actual knowledge and the actual clinical care that we have. And this is the whole idea. Regarding dysbiosis, where we have been working within genomics for a very long time, trying to demonstrate the existence of endometrial microbiome, even the existence of an endometrial microbiome in early pregnancy, which is quite different of the one in miscarriage in the very same patient, and this is published information that I just presented yesterday. We also work on trying to find out the association between a specific endometrial microbiota signature with reproductive outcome. Lactobacillus dominant is the normal one when everything goes well, but when we have this dysbiosis and you have another pathogens such as Gardnerella, such as Streptococcus, then things goes wrong. So we have found out the association between endometrial dysbiotic microbiome and reproductive failure. And just very recently came out the very first interventional study from a Japanese group that by doing the diagnosis with EMMA of endometrial microbiome, they were able to treat those patients. And when we compare control versus treatment, after personalized treatment with the specific antibiotic for those pathogens, they improve pregnancy rate, implantation rate, not only at the first transfer, but also cumulative pregnancy rate. So this is the whole range that how dysbiosis, how the microbiome may be another angle to improve implantation and clinical care in those patients with recurrent implantation failure. This work was done in recurrent implantation failure. But as I mentioned yesterday, we have to go further than that, because the silent pandemic, it is the antibiotic resistance. So there are other ways to treat microbes and to improve the situation without using antibiotics, and this is what we are working currently to try to find out at what extent we can just understand the signals that the pathogens they are sending to the human receptors, GPCR ligands, that may contribute to this whole misunderstanding of our receptors when a pathogen is there. And this is something that will be for the future. And regarding endometrial receptivity, which is the next part, first the embryo is there, and so the microbiome, an altered dysbiotic microbiome can affect the embryo and can affect implantation. But the second part will be when the embryo adheres to the endometrium. And this is just the endometrial receptivity, and this is just the window of implantation that we know for a long time that exists. And this is a personalized one that takes a limited period of time, about 36 hours, and it is perfectly demonstrated at the morphological level, but the plasma membrane transformation, the change of the cytoskeleton, allowing that this barrier allows the embryo to attach. And this only happens in a specific time of the menstrual cycle. And in fact, this is the reason why the whole menstrual cycle is about, to prepare this window of implantation. We were able to really target what is the signature, the transcriptomic signature that indicates that in one patient, the window of implantation is there, and whether the window is when we expect it, or could be delayed, or could be advanced. And we also demonstrate that at the single cell level by having single cell RNA-seq showing exactly what are the genes that are turned on and turned off at this specific time. Clinical studies has been done since then. We know that it's a controversial issue because some papers demonstrate that it works but at the end we have to find specific indications as everybody else. So nothing is for everything, and every innovation has hopes and hypes. Every innovation has the first is a panacea, then is a hell, and then at the end it gets consolidated. Look what happens with PGTA, that after 30 years now start to be incorporated in the United States. 50% of the cycles they are doing PGTA, but some countries in Europe still is forbidden, is not even allowed. So scientific innovations takes time. ERA is 10 years old, and what I can tell you from the actual published literature, meta-analysis, 2 meta-analyses published, the last one, human reproduction, very recently, is that the ERA, it is when you use for patients with recurrent implantation failure, personalized embryo transfer, improve pregnancy rate in those patients with implantation failure, whereas in patients with good prognosis, this effect has not been yet demonstrated. Therefore, I think that we are looking forward to find the specific indication for the specific type of patient. At the end is personalization. And there are more research that is ongoing now on decidualization, trying to find out the next step, which is invasion, and what is the relationship with preeclampsia, with preterm birth. So all of these things are ongoing. As you know, I am a fan of the endometrial factor. This is how we work with in my foundation. This is the main focus, just that the endometrial factor will not be a problem anymore for patients to become pregnant. So I think that this is this could be it, because I don't want to take longer time for
I mean, you make my life easy, so I don't have any questions so far. There's any question on the audience? Okay, so I think that we cover most of the parts of the endometrial factor, Carlos. We have guidelines coming from Azure regarding the endometrial factor, and what should we do? What do you think we should do? We have a key opinion leaders meeting back in, it was in April or May, and we were talking and discussing when we should apply some of the tests. So what is your thinking about it? You have your patient in front of you. So because there's 2 different things. You are clinicians, some of you, and you know that there is a guideline, but also there is a patient in front of you. So what would you recommend to your patients to perform?
Well, first of all, you have to be sure what you are doing. As a doctor, what is true is that guidelines they are running behind what is because it takes time really to create guidelines. So it is fine. You have to have them, but you have to know what is the situation, and you have to make your decision according the personalization of the patient in front of you. Again, according to guidelines, PGTA is not useful in some countries, and so I'm just to make an example. At the end, the data are there. In our world, in our scientific world, the good thing is that everything is published, and everything can be just checked and can be confronted. and everybody has to take their own decisions. But my message will be that there is no treatment for everybody, and the issue to transfer embryos at a specific day or to do a specific for everybody just doesn't work. We have to go to personalized medicine with diagnostic tests that they have biological plausibility. Statistics are fine, but you have to have a biological plausibility to do statistics. And I think that we should improve the results in our patients, because still we have this 40%, 100% is impossible in biology, but I think that we can do better than this 60% that we can have now by transferring euploid embryos. And you can look for more genetic issues in the embryo, but maybe if we look for also something in the mother, we will be better off to improve the results. But this is my own recollection.
Okay, so putting the time of the endometrio, I think that if I might request Carmen to come here, because I think that we have the two players, right? The endometrio
and the embryo.
And the embryo. So how are we gonna play together?
For sure, we are very lucky because we can test the embryo, we can test the endometrium, and what we have learned during many years of testing embryos is that not all euploid embryos implant. There is still room for improvement. We cannot be so naive to think that just having the good embryo is enough. I think we have good incubators, good culture mediums. We need to have good embryos in the lab. We need to have good selection tools for the embryo, but we need to replace the embryo in the right endometrium.
I cannot agree more. So.
So do you want to say anything else, or?
I think that I cannot agree more on that. I mean, in any process that there are two players, both of them should be considered. Try to do this with your normal life and you will see what happens. So both parts should be considered, and they're important and should be placed in the right place in the right moment.
So embryo is important, but
sense.
endometrium matters, right?
Of course.
Okay, there's any question on the audience? No? So thank you very much, Carlos and Carmen.
Thank you.
And thank you all for being here today, and see you in the next session of the Vitor Life Group Academy study. Thank you.